Hello and welcome back. Classically, the presence of a genetic component would have been demonstrated in twin studies, and therefore we need to understand the two kinds of twins. The first type of twins are the monozygotic twins, as you can see on this side of the board, and the advantage of monozygotic twins is the fact that they have identical DNA. In other words, they are clones. And this is because there was one egg, one sperm leading to one fertilization event, and the embryo during the course of development would split and develop into two individuals that have exactly the same DNA. On the other hand, the dizygotic twins or non-identical twins have the differences in their DNA as would have been expected between any two siblings, because there were two fertilization events leading to two individuals. And essentially, we can compare monozygotic twins to dizygotic twins in different kind of circumstances, about which I will tell you in a second, in order to basically demonstrate the genetic component. So the critical part in the demonstrating of the presence of a genetic component is in being able to tease apart the effects of genetics versus the effects of the environment. And so, in order to demonstrate the presence of a genetic environment, a scientist would have to compare monozygotic twins that are reared together versus dizygotic rears that are reared together. And in this case, because they are reared together, then presumably the environmental exposures would be the same or similar, but there will be a difference in the DNA. So we vary the genetics, but we keep the environment constant. And so the prediction would be, in this case, that monozygotic twins reared together will have stronger correlation than dizygotic twins reared together. That would show the presence of a genetic component. On the other hand, we can use the same kind of methodology to demonstrate the presence of an environmental component. In this case, we would be comparing monozygotic twins reared together versus monozygotic twins reared apart. Because we are using monozygotic twins in this kind of study design, the genetics is the same, the DNA is the same, but we vary the environment. And so if we see differences in these two groups, then that suggests the presence of an environmental effect. And the prediction would be that monozygotic twins reared together should be more similar than monozygotic twins reared apart. To back up my presentation, I have included some actual data of the twin studies looking at variable abilities, and scientists have been looking at the correlation of variable ability within the monozygotic twins that are reared together, and in this case, the correlation coefficient is quite strong, it's 0.76, and these were compared to dizygotic twins that are reared together, and the correlation was 0.43. And because the strength of correlation is stronger in monozygotic twins that are together than dizygotic twins that are together, that demonstrates the existence of a genetic component. And interestingly, the same trait, variable ability, also shows evidence of an environmental effect, because when we compare monozygotic twins reared together versus monozygotic twins reared apart, again, the monozygotic twins have significantly higher correlation than those that are reared apart. And so this demonstrates the presence of an environmental effect. And so this is an example of a trait that has both the effects of genetics and effects of environment. However, as you can see, the genetics component seems to be stronger in this case than the environmental, because of the differences between 0.7 and 0.4 is greater than the difference between 0.7 and 0.5. And this concludes our section on twin studies, which was the typical approach or classical approach to demonstrate the presence of a genetic component. Thank you very much.